Hello, I will be discussing the forensic application of the process in our case study. In this case study, forensic entomotoxicology was applied. It is the analysis of toxins present in a decomposing corpse via analysis of fly larvae, also known as maggots. Toxins can include but are not limited to drugs, pesticides, chemicals, funguses, plants, and environmental pollutants. This technique is seen as useful when standard toxicological procedures are difficult to use in cases where post-mortem decomposition and animal scavenging has caused blood and other specimens to not be as useful for toxicological analysis. The scientific principles behind forensic entomotoxicology is the combination of forensic entomology and toxicology, relying on insect colonization patterns, metabolism of toxins, and stability of substances. On this page, we're going to talk about analysis in forensic entomotoxicology. The insects are collected from their body during the investigation and or immediate surroundings since they may provide additional clues. They're then preserved in containers with ethanol or another preservative and are labeled with important information such as collection location, date, time, etc. The insects are identified to species level since different insect species have specific patterns of colonization or decomposing remains. After identification, certain samples are used for toxicological analysis. The samples are taken from the gut where the undigested contents may contain traces of toxins. Extracted compounds are subjected to analytical techniques such as the ones used in the case study I'm going to talk about. Those techniques are liquid chromatography, tandem mass spectrometry, and liquid chromatography quadrupole time-of-flight mass spectrometry. They are used to provide detailed information about the presence, type, and concentration of the toxins found. The results of the toxicological analysis can help establish a timeline of exposure, link suspects to the crime, and or provide insights into the cause of death. Now I'm going to discuss the analysis of forensic entomotoxicology in the case studies in our article. In case 1, larvae were collected from the stomach, upper thigh, chest, and head, and from the left eye and surrounding area in case 2. The larvae were killed by freezing at -20 degrees Celsius for at least 1 day, washed with deionized water, and dried on a filter paper along with samples of hair. The article only recognized larvae as a species. For sampling for analysis, samples were taken from larvae in skeletal muscle, liver, and lung tissue. For extraction, the samples were subjected to a protein precipitation before analysis. The liver, skeletal muscle, and lung tissue were first homogenized with 10 milliliters of isotonic sodium chloride solution for 5 minutes. Homogenization of larvae was performed with larvae samples being placed in 2 reinforced vials and adding either 100 or 200 milliliters of isotonic sodium chloride solution. The analytical techniques used were the ones I mentioned earlier: liquid chromatography, tandem mass spectrometry, and liquid chromatography quadrupole time-of-flight mass spectrometry. Lastly, the data interpretation of case 1 showed that the victim died of an overdose of fentanyl, while case 2 victims was in intoxication with the opioid methadone.